What experiments can be used to elucidate metabolic pathways? Published by sites.google.com. Chapter 17. Metabolism. Table of Contents. Introduction. Is metabolism similar in different organisms? What can be learned from metabolic maps? How do anabolic and catabolic processes form the core of metabolic pathways? What experiments can be used to elucidate metabolic pathways? What can the metabolome tell us about a biological system? What food substances form the basis of human nutrition? What experiments can be used to elucidate metabolic pathways? 1. Fermentation of glucose in yeast. Eduard Buchner, late 19th century, showed that fermentation of glucose in yeast cells yielded ethanol and carbon dioxide. This led to a search for intermediates of glucose breakdown. Metabolic inhibitors were important tools for elucidating the pathway steps. Mutations also were used to create specific metabolic blocks. 2. The use of inhibitors to reveal the sequence of reactions in a metabolic pathway. A. Control. B. Plus inhibitor. C. Observations and conclusions. Intermediates upstream of the metabolic block, B, C, and D, accumulate, revealing themselves as intermediates in the pathway. The concentration of intermediates lying downstream, E and F, will fall. 3. Isotopic tracers can be used as metabolic probes. Metabolic pathways have been elucidated by use of isotopic forms of elements. Metabolic substrates and intermediates can be labeled with a measurable isotope and then traced through a series of reactions. 2 types of isotopes have been used in this way. Radioactive isotopes, such as C and P. Stable, heavy isotopes, such as O and N. In glycolysis, if glucose was labeled with C at carbon number 6, what carbon of pyruvate would have the C? It will be at the methyl carbon. Popular isotope tracers. One of Melvin Calvin's early experiments using a radioactive isotope as a metabolic tracer. 3-phosphoglycerate, PGA, is labeled when algae are incubated with radioactive CO2. 4. NMR spectroscopy is a non-invasive metabolic probe. The nuclei of certain atomic isotopes have magnetic moments. Such nuclei can absorb radio frequency energy in the presence of a magnetic field at a unique resonant frequency. The nuclear magnetic resonance, NMR, absorption is influenced in predictable ways by the chemical nature of its neighboring atoms and by its dynamic behavior, motion. For these reasons, NMR signals can provide a wide range of structural and dynamic information about biomolecules. The metabolism of a living subject can be observed in real time with NMR spectroscopy. 5. Metabolic pathways are compartmentalized within cells. 1. Fractionation of a cell extract by differential centrifugation. 2. Compartmentalization of glycolysis, also known as the citric acid cycle, and oxidative phosphorylation. Question-related statements. Either radioactive isotopes or stable, heavy isotopes can be used to trace the course of metabolic reactions. NMR spectroscopy is a non-invasive metabolic probe. NMR spectroscopy and mass spectrometry are the principal tools of metabolomic analyses.